Hello again, and we will continue our lesson. Let me show you what you got, and you guys probably talked about it in class. And so let's verify that. Well, this is a eukaryotic cell because we have a nucleus. And so we're going to put our DNA in our nucleus, and we're going to be able to copy our DNA into messenger RNA. And so you should have put your RNA strand into your nucleus, and that is called transcription. So we're making DNA, we're going to copy that into RNA, and then this is going to move to the cytoplasm. And so I gave you two copies of mRNA because of that. So we're going to show that the mRNA goes to the cytoplasm, and then we have a ribosome. And you probably remember that from our cell unit, the ribosome makes proteins. And so now we're going to build our polypeptide using our information with RNA. We're going to build a protein, a polypeptide, with the help of the ribosome, and that was called translation. Now, in eukaryotes, transcription takes place in the nucleus, and then translation takes place in the cytoplasm. So that's what you should have found. So before we move on, we need to establish what RNA is. Now, RNA is similar to DNA. In fact, it's very similar. It's easy to get these mixed up, so fair warning. So RNA is single-stranded. Now, for mRNA, it usually doesn't tangle up, but other RNA that we're going to learn about can fall back on itself and double up and sticks because of hydrogen bonding. So you can see in this picture, it falls back in the hydrogen bonds and sticks. If you've ever wrapped a present where you get Scotch tape, the transparent tape, and then you looked away, and then suddenly your Scotch tape doubles back and sticks to itself, it's the same idea. And so RNA often makes these wacky shapes because it can loop back and bond to each other. And that's actually a pretty cool area of research that they do. And so RNA is single stranded. It is also made of nucleotides. We just call them RNA nucleotides. They have ribose instead of deoxyribose, thus the name ribonucleic acid. They also have four bases, and they're almost the same. They have A, C, G, adenine, cytosine, and guanine, but mRNA has uracil instead of thymine. Now, here's where a lot of in the past, a lot of students will often kind of try to come up with all fancy ways of trying to remember this, and I try to keep it as simple as possible. In RNA, where you would like to put a in RNA where you would like to put a T, you would put a U. So just think, oh, I normally want to put a T here, but I'm going to put a U. It just substitutes. So instead of thymine, you use uracil. We will be practicing this a lot, so don't worry if you don't get it or mix up in the beginning. I promise you, we will practice. So here's a handy dandy, nice picture to compare. So DNA has deoxyribose, RNA has ribose. DNA has thymine, RNA has uracil. And again, you do not need to know like what these actually look like. You'll see how you need to apply it. DNA and RNA both have adenine, cytosine, and guanine. They also both have phosphate. So that is, those are some of the similarities. They both have phosphate. They both have the adenine, cytosine, and guanine. And they are different in the number of strands. DNA has 2, RNA has 1. DNA has deoxyribose, RNA has ribose. Now, so let's actually take a look at how we make a copy of DNA into RNA. Remember, this is transcription. We want to make a copy of a particular gene. So we're going to use one strand of DNA. We're going to unravel the double helix. It's almost like just opening up the double helix. And we're going to use just one strand, and that strand is called a template strand. And if you've probably heard of this word before, but let's make sure. A template is something that you can use to build something. In art class, you may have used a template to draw a circle. Or in math class, you may use a protractor to make an angle. A template can be used to draw certain shapes. And so you kind of use it to build a copy. And so we're going to use, in this case, we're going to use the bottom DNA strand, the template strand. And notice our first three letters of the DNA is TAC. Now notice, in order to build our RNA, we're going to use nucleotides that base pair. So the base pairing rule still applies. Just remember, wherever you would like to put a T, put a U. So notice our first three DNA, TAC. Notice what they did is they base paired T with A in DNA would normally pair with T, but replace it with U, and we have uracil. So AU, the third letter is C, so we're going to replace that. We're not going to replace, we're going to put G. So our first three DNA, TAC. For our RNA, we're going to base pair AUG. And our second three, TAG. Well, notice T pairs with A pairs with normally T, but we're going to replace it with U, and then G pairs with C. So that's how we make an RNA strand. We use the DNA strand and base pair the correct nucleotides. And then notice once we're done with our mRNA strand, it'll fall off and we have a single strand of RNA. So why don't you guys look at this template strand over here, ATT, GCA, TAT, GCA, ATC. And why don't you guys hit pause and see if you can figure out what the mRNA strand would be for that. All right. Well, if you did that, let's go over that and see if you guys are correct. All you bright smarties here. A would normally pair with T, but this is RNA, so we're going to put a U. T pairs with A, T pairs with A. Comma, I like to use commas, that helps me visualize it. G pairs with C, C pairs with G, A would pair with T, but this is RNA. All right, and then now we have a tricky one because we got a lot of T's and A's. So let's take a look at this. T pairs with A would normally pair with U, and T pairs with A. And so that should be our third section. And then our next one should be C, G, U. And then the last one you should have found U, A, G. Now, it's always helpful to go back and double check because it is easy to make mistakes. You will get it, but it's just one of those things that your mind just flies through it. So that's what you should have found. UAA, CGU, AUA, CGU, UAG. So what do we do with this information? Well, let's step back and remind ourselves what we're trying to do. Remember, I started with this weird idea of Santa's workshop. And remember, at the end of Santa's workshop, you want to build toys. What I said was, in the cell, what you want to do is build a protein, a polypeptide. Now, how do we make a polypeptide? Well, if we go back 3 months ago, we learned that polypeptides, proteins, were made of amino acids. So that's something to remember. Proteins are made of amino acids. Now, let me remind you, you do not need to know these different types of amino acids, but I wanted you to just remind yourself, here are all of the different amino acids. Glycine, alanine, they've all got weird names. Valine, cysteine, proline, phenylalanine. So all of these different amino acids, we want to connect them together. But how do we go from ATT, GCTA, how do we go from nucleotides to amino acids strung together? It's one thing to say, oh, well, Santa has his instructions and the elves use the instructions, but how do we do that? And so, what we need to know is that there is a code. There is a code to this. 3 nucleotides make a codon, and a codon represents an amino acid. So remember I said that we used to know like how to send a secret code. Well, the secret code is 3 nucleotides form what we call a codon, and that codon means an amino acid. So how do our cells know to build this sequence of amino acids and not this sequence of amino acids? And it has to do with this code. Now, on Tuesday, we'll learn about how this happens physically, but let me let you in on the secret code. It took a good 6 years to break this in the 1960s. We had to painstakingly figure this out. So let me show you how to figure this out. So here's a codon AUG. So here's how to use the codon chart. You have the first letter A, and so where that A is, you can find all of these in here. But that's way too many. How do we eliminate that? Well, the second letter is U. So you go up here to the second letter, and here's U. And you know that you're going to go down, and where they intersect is all of the possibilities. So now we have 4 possibilities. So then we go down to the third letter, and we find our G. AUG is the amino acid methionine. Now, a lot of times you can abbreviate these, so you see met or meth from methionine. Now that also says start. We'll figure out what that means in a little bit. So AUG is methionine. Let's do CCC together, and then you guys can work together as a class. C, we're going to do our first letter, C. That means all of these possibilities here. But let's find our second letter. The second letter is C, and then you go down to where they intersect, and it's these 4. And then you go to the third letter, C, and then you go over, and it's proline. So why don't you guys give it a shot, see if you can find out what CCA is, and then AUG, CUG, GGC, UUC, and let's see if you can figure out how to break the code.